What we think is the ideal use of organoids is this patient stratification. So it's basically to really use its characteristics as a patient-like sample that you have in the lab to look at a bunch of patients, at a group of patients, and predict how that group will respond when you move your drugs forward in a clinical trial. So from our perspective, something that we can already do, but is a conceptual change on how we do drug development, is really to start using it as this patient in the lab, seeing it as a single patient or as a group of patients, and use the organoid as a platform like this, more than as a traditional sort of fancy HeLa cell.
We haven't, had a chance to look at this in animal models before. So cell therapy, CAR T, all your T-cell antibodies, the whole future of medicine is very alive per se. And so these models let you screen that really, really well.
One of the exciting things that we see in our models is that we see that the chemotherapy sensitivity and radiation sensitivity of our human patient organoid models, it really correlates or associates with what we see in the patient. Patients who have sensitive organoids that respond to the chemotherapy, they live longer without having disease recurrence. So that's a big kind of thing that gets us excited, that these may help us kind of infer or predict what's going to happen on a treatment basis level, where we can test new drugs, new therapies, new strategies to bring into our clinic.